Assalamu alaikum students. So today we are going to discuss about the organization of the globin genes. As we know very well, globin is a part of hemoglobin chain, and hemoglobin is a basic protein in our body who's carrying oxygen, and it is also the most important part of our complete blood profile. So why is it important? Why we're discussing about the globin genes? The reason behind to understand that disease is resulting from the genetic alteration in such structure. So that was necessary to grasp how the hemoglobin genes would direct the synthesis of different globin chains. So therefore we are discussing that, and it leads to understand about the hemoglobinopathies also. So here we are. This is the basically first part, which is basically alpha gene and beta gene family and steps in the globin chain synthesis. So as you can see on the screen, that part is basically, this was the basically alpha globin genes, and this was the sigma portion, alpha 2, alpha 1, and this was related to the hemoglobin, fetal hemoglobin. This was the fetal part, which is basically formed in the fetus during pregnancy, and HbA2 and HbA, and hemoglobin goer, which is a very initial part of the hemoglobin. So alpha globin gene is present on the chromosome 16, as they indicates all the chromosome 16, while the beta globin genes are present in the chromosome 11. So hemoglobin basically formed by the alpha and beta globin gene. This was the common structure, and these are the other genes which were related to the formation of mature globin genes. So two copies of alpha globin gene, the one alpha 1, which is indicated with the green, alpha 1 and alpha 2, are basically each provide alpha globin chain combined with beta globin chain. So this was very important because these all structures are very much related in that, and the kind of the epsilon gene that is expressed as in development as a component of embryonic hemoglobin, which is a HbF. And there was also some sort of other genes. Basically, globin gene families are also contain some globin genes that are expressed, and genetic information is not used to produce that globin genes. These are basically called pseudogenes. This was not produced there, but basically pseudogenes are the important part of our genetic alterations. So other one is also the, this was the alpha globin gene, and this was the beta globin synthesis, or is the final synthesis of the protein. This was the basically final synthesis. This was the genetic modification on the chromosome, while this was the like the major steps of the protein synthesis, the hemoglobin protein synthesis, which is the transcription and translation. This was very important. Introns and the exons, basically these are the two parts of the gene. And what happened with that, during the transcription, the mRNA precursor, which is a small molecule, is formed, and the splicing occurs. Splicing means they just roll over on that, and after the splicing the translation occurs and the mature protein is formed, which form outside the cell in the cytosol or in the cytoplasm. And messenger RNA is the basic precursor helping him to form the mature protein. So resulting mature mRNA enters the cytosol, where its genetic information is translated and producing a globin chain. So summary of this is shown in these both of these figures, and there was a more detail of description of protein synthesis is basically you can see in other videos. Thank you so much. Take care and enjoy your learning with basic concepts.